Good morning, everyone. Thank you for being here. So today I'm just going to be telling you a few of the results so far from my PhD. And as was mentioned, I'm from Stellenbosch University in South Africa. So we're staying in the Southern Hemisphere, we're just moving over to Africa. And my study is on the seagrass Zostera capensis. And this seagrass is a dominant seagrass in South Africa. And interestingly, it's found all the way from the west coast, where it's mainly found in the lower reaches of estuaries, all the way along the south coast and up the east coast into Kenya. And in this region, it's found more in sheltered bays. And for those of you who aren't familiar with this area, we have a really interesting dynamic with the cool, nutrient-rich Benguela current on the west and the warm, nutrient-poor Gullis current on the east, and this mixing zone on the south coast. And this is a really fascinating opportunity to study genomic environmental interactions. And this species was rated as vulnerable by the IUCN due to the general decline of seagrasses, but also because its distribution along this range is quite fragmented. And this rating makes sense in South Africa, particularly because the National Biodiversity Assessment found that 80% of our estuaries are critically endangered and under threat of losing their structure and function, represented here by these red points. And it's even more worrying because the majority of our estuaries are poorly protected, and even when there's some level of no-take protection, it's never really enforced. So there's been various genetics or population genetic studies on seagrasses globally, and they found that the diversity and connectivity of seagrasses are very context dependent. So we get low diversity and low connectivity in some areas, with clones reaching, one clone can spread kilometers. And on the other hand, we have highly structured or highly diverse populations at various spatial scales. But seagrasses in developing regions are still understudied. And in areas like East Africa, seagrass studies tend to focus on physiology and biology and less on genetics or genomics, which we know and which Catherine pointed out is very important for resilience going into the future, especially with climate change and environmental pressures. And on top of this, aquatic plant studies on focusing on phylogeography and also estuarine species in general are lacking in compared to studies on like commercial fish, for example. So with we all know the importance of seagrasses for ecosystem services, and in order to conserve these ecosystem services into the future, adaptive potential is really important in the face of climate change and environmental pressures, as this lends resilience to the seagrasses, and the traditional management plans that are often formed based solely on environment or species abundances often don't capture the evolutionary patterns behind the patterns of biodiversity that we see in our environment. So we need to look at integrating genetic diversity into these management plans. And there's an example of this in South Africa on the west coast by Nielsen et al. And they found that when you add genetic diversity metrics into conservation plans, it highlights priority areas that weren't highlighted by traditional plans. And if you exclude these measures, then you lose out on some of the processes behind the genetic patterns, and you lose out on, in a sense, safeguarding these populations against future changes. So in order to carry out my study, I looked at these 12 sampling sites. So I tried to capture the whole range of the species, but unfortunately we only have one sample from Kenya and one from Mozambique to cover the East African coast, and most of my study focused on South Africa. So I used 30 samples per site, and after extracting the DNA, we pooled the sequences per site to look at a pooled approach, because we were asking population level questions. And the sequences were sent to the Hawaii Institute of Marine Biology, where they used an EasyRAD sequencing approach to get the genomic samples. And after many data processing steps. I was able to calculate various measures of genetic diversity, differentiation. I was able to look at loci that are potentially under selection. And then I was able to integrate all of that into a conservation plan using the prioritization tool Marksan. So into the results. Here, I just want to point out from this big table the extremely low levels of diversity and the indication of a high level of inbreeding. And this leads us to believe that there's a high level of clonality in the species, and that they may rely largely on vegetative fragmentation and vegetative reproduction. And this makes sense because in this area, for the species, we've only seen flowering once under very specific lab conditions, and flowering is not actually recorded, especially in South Africa, in the wild. Here we can see the green circles represent the number of shared SNPs along the coastline, so single nucleotide polymorphisms, and then the number of private SNPs in purple along the coastline. And just take note that the green circles and the purple circles aren't at the same scale. But we can see here that not all populations have private SNPs, so this again points us to the low diversity that we see in the species, and that Kenya has the highest number of private SNPs by far. So this could indicate some level of isolation by distance or isolation by environment from the more southern populations. Looking at our population clustering analysis, I added this to the map, and the red points represent, the red points on the south and the west coast clustered together to form one group, and those on the green points on the east coast formed a second cluster. And because there's always something funny going on, this one green point in Langebaan here grouped with the east coast instead of the west coast. And our idea at the moment is that these conditions in Langebaan are much more bay-like and open, which is more similar to the east coast populations than to the estuaries that we find on the west and the south coast. And then if we look at this PCA of FSTs, we can see that the west coast populations, this cluster, are much more closely related together than the east coast cluster, where the populations are much more dispersed genetically. And we can see that Mozambique here is the most distinct population looking at this metric. So looking at these two clusters and thinking back to our temperature regime, we can see that the clusters split almost along the split between our temperature regimes. So this indicates that we could be seeing some kind of functional variation between these two clusters. Here we have our frequencies of our outlier SNPs. So these are loci which are potentially under selection, meaning that they are most likely involved in adaptation. And again, we can see this clustering pattern reflected in the frequencies of our outliers, with the same populations on the west coast having low frequencies of these outliers, and the east coast populations having much higher frequencies of these outliers. And this is really good evidence for functional variation and some kind of adaptation going on here, where these outliers are most likely adapting to something like temperature or salinity or turbidity, that we have differences between the west and the east coast. So moving on to my conservation plans. Here I integrated the genetic diversity, and I wanted to see how does it differ from traditional plans. So these maps show the differences between, for example, traditional plan based solely on habitat and using the lost fishing opportunity as a cost. And then if you add allelic richness to that plan, are planning units, do they remain selected? Are they added? Are they lost? So we can see that the majority of planning units, okay, I know this is a very small map, but the estuaries are very small. We can see that in blue, the majority of the planning units remain selected, but that when we add allelic richness, we have certain planning units that appear that weren't previously selected. And on top of that, if we add levels of adaptive variation, such as private SNPs or outlier loci, we see even more regions that are being selected with this adaptive potential on top of the traditional habitat maps or plans based on habitat. We can also see that when you add outliers, a lot of estuaries are lost here in gray, and this is probably because the cost just becomes too high of conserving all of these estuaries. So this estuary at the top here has a lot of adaptive potential, which is great to conserve into the future, but it becomes very expensive due to the lost fishing opportunity if you close off these areas. So some kind of balance needs to be found, and programs like this are really useful at identifying the best possible areas to conserve. So just to conclude, this species has really low levels of diversity, and this means that they're vulnerable to change because genetic diversity forms the basis for adaptation. This also means that they'll be less resilient to environmental changes, but that we might have some level of pre-adaptation to warming with our populations on the east coast showing some kind of adaptive variation that those on the west coast aren't. And then we can also see from the conservation plans that it's important to include some measure of genetic diversity and potentially even adaptive variation into conservation plans in order to make our reserves more resilient into the future. Thank you. I'd just like to thank all my funders and the conference for helping me be here.
Do we have any questions? We have quite a bit of time. Great talk. That was really interesting. I have. Is this on? Yeah. I have 2 questions. What were your estimates of clonal richness in your studies, or how many out of your 30 samples, how many individuals were unique genotypes?
It was actually really unfortunate that we weren't able to calculate that because of our pooled sequencing approach. We had one population as a single unit. So that, if this work was to carry on, that would be the next step, to really pick that apart and look at clonal diversity. So we're thinking of taking some of the SNPs that I found and creating SNP markers to then go, because it's obviously really expensive to do next generation sequencing on 30 samples for 12 sites. Yeah.
And my second question was about the adaptive loci. Were you able to blast those sequences and see what they were, if they were linked to anything known?
So I didn't include the results here because they mostly blasted to hypothetical proteins. But those hypothetical proteins are hypothesized to be linked to cell membranes and cell wall thickening. So if we look at that with our temperatures, it may be some kind of protection against increased temperatures. This is what some other papers also suggested, but it's no hard evidence and no direct links to genes where we know they function.
Thank you.
Okay.
You can ask something. Really great work, and we know how hard it is to do some of this study, so well done. Your hypothesised break around temperature, does that happen to coincide with any different currents? I think there's one of the currents that splits around about there as well. Yeah,
so that's sort of where the Agalis comes down. We have retroflections on the south coast, and there's a lot of biogeographic studies that find breaks there in a similar place.
Yeah. So another alternative hypothesis for the sample that ended up on the west coast, or the genotype type, might just be a random long distance movement. Doesn't unless you can prove there's some reason for it. That we see that kind of thing happen occasionally. Things move.
Yeah.
And then the second half of the question, I guess, is did you model do any genetic drift models to work out if the pattern essentially could also be random?
We haven't actually tried that yet, so that's a great suggestion. I'll have to look into that. Thanks.
Really interesting.
Thanks.
What actually in the E is the targets for
Sorry?
Your EZ rad,
Yes?
what it targets for?
What were the targets?
Yeah. Any locus or any markers?
No specific locus or markers, so it looks at random sections along the entire genome using restriction enzymes.
Oh, it randomly select by its own.
Sorry, can you repeat that?
It randomly select the sequences by its own.
So it's based on restriction enzymes. So wherever those enzymes cut,
Okay.
it's able to sequence those sections.
Okay.
But it's not specific, if that makes sense.
Okay. How it is advantageous when compared to another markers?
Sorry, can you repeat that?
What is the advantage of using this particular random sequences?
Yes.
Presently, many markers are there, like SSR, extinctions or RNA, many things are there.
Yeah.
What is the advantage of this?
The advantage is that you get a whole genome view,
Okay.
but at a reduced level. So we can see bits of the genome.
But for that, every species whole genome sequence should be available in the data banks.
Sorry?
If every genome's sequence should be available in the data bank, then only it can, EZRAD can work, you know. Whole genome of the every species should be available in the data bank.
Sorry, I don't know what you're asking.
No. You see, when it's targeting for the whole sequence in the whole genome sequence of a particular seagrass should be available in the data bank.
Yeah, so it's not based off of markers, but just restriction enzymes. So
Okay.
it's a de novo approach, but luckily I didn't have to use de novo assembly. I could map my results to Zostera marina. Yeah, I hope
Thank
that
you.
helps.
Can I just interrupt you there? Just so you guys know, the next speaker, Matthew, will question. So you can either duck out now and go to the another session if you like, or you can sit here in eager anticipation for the next talk.
The timing is off between the two, so maybe we can use that as a... Oh, you know that.
That's what I thought, but yeah, we also have one more question after that, so I thought, yeah, those who want to sneak out for the next one, go for it. If you want to stay, stay. But that gentleman's been for a while, so I think we'll let him answer the question.
Thank you. Salmon Bandeira from Mozambique. My question is more to link the genomics with ecology. I just wonder whether within those sites in South Africa or the one in Etinyako, Maputo Bay, and in Kenya, did you find some variability? Was that one individual or?
In the genetics or in the ecology? Sorry, you mentioned genetics and ecology.
Yes. Can you link that variability with some sort of morphometrics that are common in the area? For example, in the north, you tend to have narrow leaf type of forms,
Yeah.
although in South Africa you may have variability. I just wonder if...
Yeah, that would be a great idea. We just don't have morphometric data like that. So that would be a great idea to see how is this playing out in a phenotypic response.
Okay, thank you.
Yeah, thanks.